Good day, everyone. I am Mirana D. Sumaya, taking up Bachelor of Science in Agriculture at the Visayas State University. For today's presentation, we will tackle about cell cycle. But in this topic, we will be focusing the differences of mitosis and meiosis. Cell cycle. It is one of the most breathtaking processes that helps the cell to not only grow but also divide. We know that the cell cycle has two major phases. One helps in preparation of the cell for the division. This preparatory phase is the interphase. Moving on the second phase, it carries decision of the cell. This phase is the M phase, where M stands for either mitosis or meiosis. Before we begin, allow me to first discuss the cell cycle for better understanding. Mitosis and meiosis is technically part of the cell cycle, and so cell cycle is how a cell duplicates itself. In most of the time, it is in this orange phase, this one, which is called interphase. In G1 phase, the cell is growing, so it is getting larger. It enters in the S phase, or the synthesis phase, where it is duplicating all the DNA. Then finally, it goes to the G2 phase where the cell continues to grow. However, some cells do not undergo here because they do not copy themselves like nerve and muscle cells. So they enters to a called GO phase and never divide. But then, if the cell goes into division, it will then go to M phase, where mitosis and meiosis are dividing the nuclei. Question: how does the cell decide whether to undergo mitosis or meiosis? Well, that depends on the various factors and the type of cell undergoing division. Let us now proceed to the main topic of this report. Let's spot the differences of mitosis and meiosis. The first difference between the two processes is that mitosis occurs in somatic body cells. Example for this is the skin cells and then the nerve cell, while meiosis occurs exclusively in reproductive cells, the sperm cell and the egg cell. That means mitosis mitosis occurring irrespective of whether an individual is male or a female gives rise to a somatic cell, which are exact copies of the parent cells. On the other hand, meiosis occurs in males and females, giving rise to haploid sperm and egg cells respectively. The process of meiosis, however, has a different names in both sexes. This is due to the different cell produced as an end result. Meiosis is also referred to as oogenesis, that is the process of ovum reproduction in females, and spermatogenesis, the sperm production process in males. Another difference is that mitosis used as a mode of asexual reproduction in a lower organisms like budding, while in meiosis helps in forming gametes used as a male of sexual reproduction like plants and animals. Now, let's move into the major differences. Take a look at these images. Observing these images, we can easily notice that most basically differences between the two. Let's begin noting down each difference individually. The first on the list is that mitosis has only one cell division, while meiosis involves two successive cell division. That simply means mitosis is a single-step division while meiosis is a two-step division. And this results meiosis having two phases, namely meiosis 1 and meiosis 2. The second difference is that mitosis produces two daughter cells, while meiosis produces four daughter cells. The next major difference is that based on chromosomal number of the daughter cells. In mitosis, the daughter cells are diploid with two sets of chromosomes just like the parent cell. While in meiosis, the daughter cells produced are haploid with only one set of chromosomes. These daughter cells are called gametes that have exactly half a number of chromosomes from the parent's cell. The former difference also gives cue about one more difference between the two. The image clearly depicts that all the four daughter cells are produced by meiosis are genetically different from each other, while two daughter cells that are produced by mitosis are genetically similar with each other. Now let's move on to the next difference. But before that, let us know how many stages of cell division a mitosis is and meiosis have. In mitosis, there are four stages, while in meiosis, it involves two rounds of cell division. There are the meiosis 1 and meiosis 2. Therefore, it consists 8 stages of cell division. Now moving on to the next difference. Mitosis occurs in all organisms except viruses, while meiosis occurs in plants and fungi, as well as animals. As we go further, let's dive deeper and compare the stages of mitosis and meiosis to explore some differences between the two. The first stage of meiosis 1 is that prophase 1 is very long, consisting of sub-stages. On the other hand, prophase of mitosis is comparatively shorter than the prophase of 1. Crossing over, this one, crossing over does not occur in prophase, while definitely occurs in prophase 1. Independent assortment is technically absent in metaphase of mitosis, while it occurs in metaphase 1 in meiosis. The separation of chromatids takes place in the anaphase in mitosis, and on the other hand, the separation of the homologous chromosomes take place during anaphase 1 and the separation of sister chromatids during anaphase 2. So those are the major differences of mitosis and meiosis. Let us now summarize these in one table. Have you ever wondered what important tasks do these two processes is performed in our body? Well, mitosis helps in healing and repairing of the cells and tissues of our body. It is also responsible for the growth and development of the body. Meiosis, however, is exclusively helps in producing gametes, and these ensures genetic diversity. And with this, we come to an end of the concept of cell cycle.
The two gametes of sexual reproduction are sperm and egg. Did you notice the large size difference between the sperm and the egg? There's a reason for this, but let's see how their formation differs from one another first. The creation of sperm is called spermatogenesis. It starts with a diploid cell called a primary spermatocyte, and after meiosis I, it forms two secondary spermatocytes, which are haploid, but have a duplicate chromatid with them. After meiosis II, all the cells are haploid, which in humans means they'll have 23 chromosomes. The haploid spermatids will mature into sperm, which will then be motile and ready to fertilize an egg. The creation of an egg, also known as an ovum, is called oogenesis. Like spermatogenesis, will start with a diploid cell called a primary oocyte. But after meiosis I, the cytoplasm is divided unevenly, and one cell gets most of the cytoplasm. The cell with the most cytoplasm is called the secondary oocyte, and the other small cell is a polar body. Both of the cells are haploid, with all chromosomes duplicated. After meiosis II, the secondary oocyte is again divided unevenly, and most of the cytoplasm goes to the ootid, which will mature into an ovum. This ovum will have all the organelles, including the mitochondria. Even though only one mature ovum is created by the one oocyte in oogenesis, females are still born with millions of cells and will release hundreds in their lifetime. Not every cell created by one individual is the same. In fact, they're all very different. Genetic recombination from crossing over and independent assortment during meiosis lead to genetic variation and differences in each sperm and egg. This is why brothers and sisters don't all look identical. Now, the whole point of making sperm and egg is for fertilization, to make offspring, babies. Once the haploid set of chromosomes from the sperm are released into the egg, the two sets of chromosomes combine, creating one diploid zygote, which will grow and mature over time into the offspring. We all started out this way, just a tiny cell the size of a grain of sand. But sometimes things don't always go so smoothly. Errors during meiosis can cause non-disjunction. Non-disjunction is a failure of homologous chromosomes or sister chromatids to separate during meiosis. Here's one of those cases. During metaphase I, everything is lined up as expected. But during anaphase I, a chromosome is pulled to the wrong side, which leads to gametes with incorrect numbers. Some will have an extra chromosome, and some will have one less. This could lead to a variety of issues. While most of these cases will lead to miscarriage, some disorders are not life-threatening. Here are some examples of disorders that can occur as a result of non-disjunction. Probably the most widely known one is Down syndrome. In this case, there is an extra 21st chromosome, which can lead to mental impairment and stunted growth, among other things. Klinefelter syndrome occurs when there's an extra X chromosome in the standard male karyotype. This can lead to weaker muscles and bones, less testosterone, and sterility. One of the only cases of monosomy where there's one less chromosome is Turner syndrome. In this case, the second X sex chromosome is missing. The female will be sterile and have shorter stature and a webbed neck, but may not have any mental impairments. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on Twitter at SciencePet.
So that's end my report. Thank you for watching.